Yeah, we can see your first slide.
Okay.
So let's get started. Hello everyone, welcome to our Nanomotor mini talk. It's my great honor to introduce our today's speaker, Professor Walter Paxton, to give a talk on our mini talk. Professor Paxton did his PhD at Penn State University. I believe we're all familiar with his significant work about Catalytic Nanomotors, which is published on JACS in 2004. Now he's an assistant professor at Brigham Young University. His research specialized in 4 areas, including soft nanotechnology, designing and controlling the self-assembly of stimuli-responsive polymers, turning the chemical and the electronic properties of interfaces, and integrating biological function in stimuli-responsive vesicles and bilayers. And today, he will talk about a brief history of catalytic nanomotors. So without further ado, let's welcome Professor Paxton to share his view about nanomotor. So please go ahead, Professor.
All right, thank you very much. So this has been a lot of fun to kind of go back through the archives and find some old papers, some old things that we'd written and done through this period between like 2001 and 2006 while I was a student at Penn State. We started by thinking a little bit about motion and how motion is different at the small scale compared to the macro scale that we're familiar with in our daily experience. So on one side, you've got some of the largest living things, the dinosaurs, the megasauruses, and then on the other side, you've got E. coli bacteria that can swim around, and then we're kind of somewhere in the middle, but the physics that dominate under these different scales can be significantly different. This isn't something that we completely appreciated, I think, when we started doing this, but I want to make that point right off the bat that that the scaling of physical laws is going to be different for different phenomena. And so this miniaturization of things has been a pretty popular topic in entertainment, from the Fantastic Voyage in the 60s, this early movie, to Honey, I Shrunk the Kids and Inner Space in the 80s, and then Gulliver's Travels featuring Jack Black in the early 2000s. And all of these kind of looked at the small world, and most of them are looking at mapping what our experience is in the macro scale down to the micro scale. In a really interesting perspective article, The Size of Man in the American Scientist, 1968, it talks about some of these scaling laws, and he talks about John Swift's book, Gulliver's Travels, and he says that some physical properties of a body are proportional to the linear dimensions, and others vary with the third power of linear size, such as weight and cell number. And then he kind of praises Jonathan Swift for getting some things right and pointing out some things that weren't quite so right. So the size of humans matters a great deal in our intuition about things, and sometimes we need to break that intuition, and some easy ways to do that are to look at things like insects that can crawl upside down, where their van der Waals interactions between their feet and the surface can overcome or the force of gravity so that they can walk on walls in inverted structures. You've also got electrostatics, which can overcome gravity by having electrostatic repulsions. If you build up static charge, your hair tends to stand on end in opposition to the gravity, which would normally want to push that down. Surface tension is another phenomena that involves the intermolecular forces of different substances. And so here you have a water strider that is able to stand on top of water instead of being entrapped in the water or submerged due to gravity. And then surface tension also has some really interesting effects. So you're going to see a lot of videos here that I've collected over the years, and this is an ant drinking something, and you can see that that bubble there is not like in a cup; it's just a droplet because of the surface tension. And so the size of people also affects how matter is transported in the body. you think about single-celled organisms, they can rely on diffusion just within the cell, but the human body as a collection of cells all over, it has to rely on other transport mechanisms to get nutrients to cells and to remove waste products from cells. It has to have some other mechanisms. And so, so we've evolved circulatory systems to help with the transport of material in the body. All right, so we were thinking about micromotion, and we were thinking about ways that we could move small objects, and we were inspired by some other phenomena observed in nature. In 1972, Peter Mitchell, who won the Nobel Prize for chemoosmotic transport in cells, he described a mechanism by which flagellar bacteria could move. And this was at the time, we didn't know what the flagella were for, but we knew that they were involved in motion. And so he proposed this idea that if you have a cell that pumps protons out at one spot in the cell, and then those protons are able to come back in on the opposite end, say at the end of a flagella, then this could give rise to a self-electrophoretic motion of single-celled organisms. Howard Berg was actually one of the guys who looked at cell motility probably more than anyone in the world. And in 1973, he was looking at E. coli bacteria and observed this. And this didn't look necessarily like some kind of static tail that was allowing for ion migration to drive the propulsion of these single cells. And so he actually did a really cool experiment where he took these E. coli cells and then he bound them to a surface by their flagella. And what he observed was pretty striking. These tethered cells, now instead of the cell itself moving, or the flagella propelling the cell through solution, what you have is the cell stuck on the surface and then the cell body is rotating. So this indicated that there was some rotary motion, which made this explanation of Mitchell's a little obsolete. And so now we have this understanding that these flagella are rotated by protein machinery that are embedded in cell membranes, and that gives rise to the rotation of this flagella, which allows it to have propulsion, sort of like a corkscrew mechanism, and utilizing the viscous drag of the solution around it to propel it through solution. So there's other examples of biological micromotion, and this is a white blood cell. Dave Rogers at Vanderbilt recorded this in the 1950s. This is a white blood cell chasing down a pathogen, and the mechanism for motion here is understood to be a cytoskeletal network that is being reconfigured, where you have a leading edge that is pushed along by these growing microtubulin or actin filaments. And then eventually this white blood cell gets its prey and takes care of business like it's supposed to do. Some kinds of bacteria have found a way to hijack that mechanism. And so this is Listeria bacteria that's responsible for some kinds of food poisoning. And it's thought to take advantage of this actin polymerization and propel itself through solution or through a cell. And you can see that it does so with maybe some pretty nasty effects to this host cell. But these things move around pretty good. And so this was, these videos were taken, this is Julie Theriault at Stanford. And they actually mimicked this by taking the protein machinery responsible for the actin polymerization and putting it onto a non-living polystyrene bead, and took these really nice SEM images which show this growing filament coming from the end of a polystyrene bead. And so this showed the motion of, this could be used to drive the artificial motion of polystyrene beads. And this was 1999. So this is before we'd started doing anything. And this is taking biological machinery to do this. So some other examples of harnessing biomechanical systems that kind of preceded what we were doing was this use of the F1 ATPase, which is responsible for converting ATP to AGP to ATP. Wait, what is it? ADP to ATP by taking advantage of transmembrane proton gradients. And in addition to generating more ATP, these proteins were also demonstrated to be rotary proteins. And so there's a rotation that's involved as this catalytic process proceeds. So using this, Montemagno, who was at Cornell at the time, immobilized these proteins onto some microfabricated devices and then tethered some nickel to the top of those proteins, and then showed that these proteins could drive the motion of these microscale nickel bars, which was sort of a miniaturization of a hybrid macromechanical device where you drive the rotation of something with some beast of burden to do the work for you. This is happening on the micro and nano scale. Another example of this that was coming out late, well, it started in the 80s, but exploiting this phenomenon in the 90s are these linear motor proteins like kinesin and dynein. And this was the Ron Vale's lab in 1985 and 1988. They had observed the movement of dynein proteins. And so this is a filament. And if you watch this, you can see the motion of these dyneins along the microtubule filaments. And so kinesin was another motor protein that does the same kind of thing, which was really amenable to being immobilized onto surfaces. So the Vale lab developed an inverted kinesin experiment where they took kinesin, it's immobilized onto a surface, and then they have fluorescently labeled microtubulin that they can watch move across the surface of these. So the Vale lab did this in 1988. I couldn't find any of the original videos. This video comes from the Koch lab. You can see the biocatalytic activity driving the motion of these microtubulin filaments on surfaces. So what about wholly synthetic nanomotion? Something that doesn't involve a biologically derived machinery. So there are a couple of examples of this. The first one was in Science in 2000. They looked at tin deposited onto copper surfaces, and they observed that this tin, this is under electron microscopy, they observed that this tin would migrate across the surface, and behind it, it would leave this alloy of tin copper. And so the dissipation of energy is the formation of this alloy that drove the motion of this tin on a surface. So this was about 2000, and then we came across this paper here. This is a solvent dissolution. These are polymer beads, and these polymer beads have some kind of organic solvent dissolved in them, and that organic solvent will leak out when these beads are put on the surface of water, and that leaking is asymmetric, and so you get a generation of a change in interfacial tension that will drive the motion of these objects. So this is where the state of the field was at the time, and to be frank, we were ignorant of a lot of this state of the field, but just for historical context, there hadn't been any wholly synthetic nanomotion driven by catalysis at the time that we started these experiments. And so that's when I joined Penn State and the SEN group in about 2001. And so I think this picture was taken probably 2004 before, but you can see some people, some names you might recognize. Ayusman Sen, of course, Tim Klein, and Shakuntala, and Eve is right here behind me. Ying Hong, who did some of the chemotaxis work. So when I joined the group, I remember asking Dr. Sen, so what do you want me to work on? And he says, I don't care what you do, just make me famous. So I took a little bit of liberty with that to think, well, we could probably do a lot of different kinds of things here. What things are interesting? So for my first year progress report, like a lot of people in the Sen group at the time, I started looking at acrylate polymerization. But I also noted in my first year progress report, the idea of nanoscale mechanical devices is interesting, like motors and pumps. And then I talked about this Whiteside's paper that I'll talk about again in a minute. They reported the autonomous movement of small 9mm diameter plastic discs in hydrogen peroxide solution propelled by catalytic decomposition of hydrogen peroxide on the bottom sides of the disc. So this was in my progress report in 2002. And I don't know why, but I still have copies of that stuff on some hard disks, on some CD-ROMs. So along these lines, I noted that all particles move autonomously because of Brownian motion. And with Brownian motion, the interaction of the particles is random and based on weak or based on random fluctuations and weak or particle interactions. So I said, well, particles that react with surroundings might have trajectories and velocities that differ from same size particles without surface reactions. Particles could then be designed in such a way as to harness these forces and cause controlled translational motion. So it was actually Dr. Sen who pointed this paper out to me, the Whiteside's paper that I'll talk about. And so we've been talking about this and having these ideas about how we could drive the motion of small objects. So this is the paper that I've been pointing to. This is these were PDMS discs that had hydrophobic and hydrophilic components in them. And they used that because they were also looking at self-assembly on the macro scale. But the key point here is that they had platinum covered porous glass, and they coated that with platinum and then put it on a aqueous surface that had hydrogen peroxide in it to release oxygen. The platinum is a really nice catalyst for the decomposition of hydrogen peroxide. So there's this dissipation of chemical energy that caused the growth, nucleation of bubbles that drove the motion of these discs. And so we were asking, well, how small can we make these? Could we do something like this on the small scale? So in spring of 2002, as part of my first year committee meeting, I had talked about making something like this, where you've got some kind of catalyst particles and then expose them to hydrogen peroxide. And you have this asymmetric structure so that you generate oxygen on one side. You'd still have the diffusion of hydrogen peroxide going in and the diffusion of oxygen bubbles coming out, and having that drive the motion of much smaller objects. So in my first year committee meeting, Tom Maluk says, hey, you know what? We can make this kind of thing in the lab right now. So let's do it. And he showed me how to do this. And we were looking at coating the insides of these aluminum oxide membranes, which have continuous parallel channels, about 60 microns long. And each of these pores is somewhere between 100 to 300 or 400 nanometers across. And so we were going to coat the inside of these with silica using a method that Nina Kovtukova developed. And she worked on this early on. And we electroplated inside of those. We electroplated some silver, gold, and then platinum. And then the idea was that we would sonicate these and then get some asymmetric structures that had catalytic regions that were sort of hidden and would provide this asymmetry that would allow oxygen bubbles to grow and nucleate and push these things along, sort of like the Whiteside's discs. All right, so this is what we were looking at now. for summer 2002. Platinum gold wrapped in silica with a short empty portion of the tube between the platinum and the end of the tube. We thought it would decomposition of hydrogen peroxide into water and oxygen would generate some kind of thrust and propel the particles forward. As it turns out, this was a difficult target. And so that frustrated us a little bit. And so, I was just looking at how to electroplate things with platinum and gold structures. And so these are uncoated structures. And although this wasn't the target structure, I decided, what the heck, we'll just put these in hydrogen peroxide anyways. So this is, as far as I believe, the very first catalytic nanomotor video. And I have this disc. This file is only, let's see, how long is this video? Like 48 seconds. And the file was something like 500 megabytes because we didn't have really good encryption and compression at the time. But you can see the motion of these objects. We've got a rotating spinner here, and then we've got swimmers right here. So the frame rate is really low here, but this is the very first one, and it's on this disc right here. And Ayusman, I think you should have a copy of this disc also. If you don't, I'll make you one. So improving frame rate and some image quality things. We started looking at things like this. So this is on the 31st. So almost, what would this be, 19 years ago almost. And I remember seeing this in the microscope for the first time and getting the very first video. And I remember specifically not telling Dr. Sen for a day or two because I just wanted to like enjoy just knowing that I was the only person on earth who had ever seen this before. And so then a few days later, shared these videos with him. And so this is the one that I think ended up in the original JAX paper in 2004. And we'll talk about that here too. But really fun to look at still, even after all these years. All right, so the catalytic motors 3.0 had no silica shell around them. And we've got gold platinum structures that are decomposing hydrogen peroxide into water and oxygen on the platinum end. And we make these by this electrodeposition technique and characterize them. And so you can see that even though we idealize these as perfect cylinders, there is some asymmetry with them. But we can also distinguish between the platinum and the gold segment by optical microscopy, which became really important later on. So one of the things that's really important is that for those who've been working on this for a while, you'll know that everything that's dispersed in fluids is going to exhibit some kind of motion if it's not fixed to a surface. And so these are pollen grains. This is similar to what Robert Brown first observed in 1827. And that same phenomena happens for these platinum gold structures if they're just in deionized water. And so if you watch these things long enough, you'll start to think, oh, hey, they look like they're moving actually. Well, and they are, but they're moving randomly and stochastically. And if you have your sample interval short enough, you'll find some things that look like directed motion. But this is not. So we did some critical experiments to distinguish between passive and active motion. To do this, I defined a couple of terms that I don't think are really critical to go over right now, but the important thing was that as we decrease the concentration of hydrogen peroxide, we notice that average speed decreases as well. But even in pure water, if we're looking at the center to center displacement from point here to point here, and then looking at that for in between each frame, it looks like these are moving, 3 to 4 microns per second. This is dependent on the time interval between subsequent frames in time. But if you look at pure water and you look at this directionality factor that we define, some kind of orientational parameter between the orientation between its initial orientation and the direction that it actually moves, we could distinguish between directed motion and random Brownian motion, where this should approach 0 as we get a large enough sample size. So even though it looks like it's moving, if we just look at the average speed, if we're looking at the axial velocity, it's significantly lower and a lot more stochastic. Okay, so that was one critical experiment to distinguish between active motion and passive motion. The next experiment was, which direction are these things actually moving? This came up in one of the questions from one of the reviewers. So in the Whiteside's experiment, the bubble propulsion, this nucleation and growth of bubbles that are pushing this along, that's driving the motion away from the platinum. So if you have a rod, well, you might expect bubbles to nucleate and grow and push this in the same direction. But when we looked at what was happening, you'll notice two things. First, there's no bubbles. And second, if we identify the platinum side, and then we watch which direction these things are actually moving, they're actually moving towards that platinum end. And that was a little bit of a surprise to us at the time. No bubbles near the motors, and the bubble recoil predicts the wrong direction, so we ruled out bubble recoil. We attributed the lack of bubbles to the fact that the oxygen concentration, even though it might be high, it has to overcome some kind of energy barrier in order to grow, to nucleate the bubbles and grow those bubbles. And so there wasn't a high enough concentration for that to happen, at least in the immediate vicinity of the first catalytic nanomotors. So what about the we would add things like surfactant or other kinds of organic solvents that would change the surface tension of water. And we noticed that those things would also correspond to a decrease in the observed velocity of these catalytic nanomotors. But in hindsight, all of these things also poison catalytic platinum surfaces and affect the reaction kinetics. So it did not rule out other mechanisms that in hindsight we started thinking about, like temperature gradients, viscosity, auto-diffusiophoresis, and auto-electrophoresis. So I settled on this surface tension mechanism, and I thought I had something pretty described. And every time I would explain it to Dr. Sen, he'd say, okay, walk me through it one more time. And I think rightly, and that turned out to be something that was pretty important. So the first paper, here's a long timeline. We submitted it after doing the initial experiments in October of 2002. We submitted it to Nature in March of 2003. April, we get a comment back that 2 of the 3 referees are intrigued by our work, but all of them raise a number of important technical concerns specifically related to the mechanistic interpretation. Reviewer 2 actually was the shortest review and said this shouldn't be published. They don't know what they're talking about. But we resubmitted to Nature. We defended this interfacial mechanism and supporting experiments with a derived model, enlisting the aid of Vin Crespi and his postdoc Paul Lammert, who further developed this interfacial tension mechanism. The reviewers were still not convinced about the mechanism. We resubmitted to Nature with more supporting evidence. I think this involved some force microscopy of nanobubbles that we saw. And then in January 2004, they finally said, okay, you need to submit this elsewhere. So after almost a year, they finally said, okay, we're not going to publish it. So we submitted to Science, and they said submit elsewhere almost immediately. So we submitted it to JAX as a communication without the mechanism. And then they said, well, we don't really understand how this is working. Can you say anything about that? And I remember saying, okay, you want a mechanism? We'll give you a mechanism then. So we resubmitted as a JAX article, again, with a plausible mechanism that was consistent with our observations. And then it was published in JAX as an article. So that was really nice. And one of the cool things that came out of this review process is somebody asked us to look at the energy efficiency of the interfacial tension engines. And so the idea is that if you have a particle moving, you can calculate what the drag force is that the propulsive force is opposed to. So if it's going at some terminal velocity, this drag force will be balanced by the propulsive force. So we did some calculations, and we wanted to be really accurate. So we looked at the drag force for a cylinder, not a sphere, as if the discrepancy would mean anything, and found that the power output was something like 10 to the minus 19 joules per second. Power input, based on the reaction kinetics and the free energy of the decomposition reaction, gave us something on the order of 10 to the minus 8 joules per second. So the efficiency of these early catalytic nanomotors is something like 10 to the minus 11 or 10 to the minus 9%, if that makes it any better. And I remember Tom saying the bad news is this is a uselessly inefficient motor. The good news is there's lots of room for improvement, 10 orders of magnitude of improvement. So that was actually a little bit of a silver lining there. And we have seen some significant improvements over the years in terms of the efficiency of these early catalytic nanomotors. But this was finally published, and we were really happy about that. And almost immediately, I was thinking, well, that mechanism might be consistent, but I don't think it's correct anymore. About the same time in Toronto, Jeff Ozen and Ian Manners had created some nickel gold structures and observed some interesting behavior. They called them synthetic self-propelled nanorotors. And it was kind of fun to see in the footnote, it says, no added improve. At the time our paper was submitted to Chemical Communications, Maluk and coworkers reported in the Journal of the American Chemical Society that catalytic nanorods with different compositions could undergo linear type motions in solution, and then cited our paper, which was kind of fun. So it was fun to be first, for sure. So we were looking at what other kinds of things can we do here? Where do we go from here? One obvious thing for me was nailing down this mechanism. What's really going on here? And then looking at things like changing the raw geometry, looking at increasing the efficiency by controlling plating conditions, and then extending the idea to other reactions, other catalytic reactions. And we were already at that point looking at using enzymes as well. About this time, we sent our first proposal to NSF. I can't remember if this was before the JAX paper was published or after, but we got some really glowing reviews. The panel summary said, original and creative, great potential, very well organized and conceived, impressive preliminary work, exactly the kind of work that will move nanotechnology forward. And that was fantastic. Every single reviewer rated it excellent, excellent, excellent. And that was really fun, except it was ultimately declined. Fantastic ideas still get turned down at the NSF, just so you know. I think the next year we didn't have any trouble and we got this funded. But I add that in there because it was one of those things that we knew we had something good and it wasn't getting the love that it needed at the time. So we did some other fun experiments anyways, with support from Penn State's MRSEC, with Lily, I don't remember Lily's last name, but she was in the Jackson group, and she created these patterned surfaces for us. And so we started putting our nanomotors on these surfaces, and they're just kind of fun to watch here. So swimming around a little bit. One of the things that I want to point out is one of these motors here. I think I'll just scroll through it because it's a little bit faster here. Okay, so if you watch this particle, can you see my mouse? This particle here and this particle here, they are on track for a collision. And so we've got these two catalytic motors kind of fighting it out. And if you can't beat them, then join them. And those two keep swimming along. So we started observing some really interesting phenomena here. And so I joked that this was the catalytic nanomotor flea circus, where we could just get them to do interesting things and put them in interesting environments to see what happens. Tim Klein joined the group in 2004 or 2005 and started working on incorporating nickel segments to these. And so this was published in 2005. This video I don't think was published, but I'll kind of scroll through this a little quickly. This is a passive particle, and this is an active particle that has nickel segments in it. And so we're basically taking a magnet near the microscope, and we're just rotating that magnet around in order to get this particle. We wanted to get this particle to crash into the passive target. And so a couple of attempts there, but just being able to control these was really fun at the time. We also looked at the effect of catalysis and asymmetry. I don't think this one was ever published in a paper either, but I do know that it's gotten out somehow. But these were particles these were rods that were gold, platinum, gold, and then some of the particles have been damaged, revealing a more asymmetric structure. And so you can, well, this one's fun to watch in real time. Oh no. All right, so I think this may have crashed. So let's see, we'll restart the program here. I mean, if we're still having problems with videos today, you can imagine the trouble that we had when we were doing all of this work. So let me come back here. Okay. All right, so I'm not going to scroll through this one. I'll just play it. And so most of these are not doing a whole lot, but this one right here that's broken is actually moving at a pretty good clip there compared to everything else, which I think really emphasizes the difference between active and passive motion there. Jeff Kaczmarek had made these platinum gold gears, which exhibited rotary motion, and published this work in Small in 2005. I know that we had a heck of a time getting this experiment to work, and I think part of the problem is that the platinum that was evaporated onto the surfaces was different, lower surface area than the platinum that we were electrodepositing. Okay, so why do they swim? We looked at a couple of different possibilities. We ruled out bubble propulsion, and then we looked at diffusiophoresis as a potential candidate for propulsive force. And this is about the time we got connected with Daryl Veligal in chemical engineering. And he pointed out this really nice paper from John Anderson from 1989, where he looked at phoretic behavior, including he discussed a little bit of autophoretic behavior, but there weren't any examples of it at the time. So the thought was that if you have an oxygen concentration gradient emanating from this, then that oxygen concentration gradient could exhibit a diffusiophoretic force along the gold end of that. But in our hands, we looked at this, and it predicted motion towards the gold and the speed of less than 4 nanometers per second for the predicted oxygen concentration gradient. So we're looking at things and we're like, well, that's not the mechanism, and that's not the mechanism. So what else could it be? Daryl Veligal, again, pointing to this paper with by John Anderson, we started to look at the potential of electrokinetic phenomena. So electrokinetic behavior is if you have two electrodes and you're running a current through an aqueous solution or other kinds of solution as well, there's an electric double layer that is formed between the surface, the stationary surface, and the mobile fluid, and the charge is in that mobile part, and that charge can be affected by applying an electric field. So applying a potential between these two electrodes drives the electromigration of the ions and the water molecules that are associated with it. And so you can get what's called electroosmosis. But if you have particles that are dispersed in there and you exhibit and you expose them to an external electric field, then these particles will also migrate due to a complementary phenomenon. Now, interestingly enough, Paul Lammert, who was one of our colleagues who helped us develop the now obsolete mechanism, had also written a paper on ion drive for vesicles and cells. And so he looked at electrokinetic phenomena for particles that were pumping protons out on one side and pumping them in on the other side. And it's kind of funny because at the time he was in France, and I was like, yeah, there's another Lammert, but he's Lamer, the Frenchman. And I thought it was a different person, and it wasn't for a while until I finally made that connection that it was the same person. And so we started looking at this potential for an electrokinetic mechanism where we have some species that's reduced gets oxidized on one side, and another species that's oxidized gets reduced on the opposite end. And we thought about how that might drive the motion of objects. And we included this in Chemistry European Journal perspective, or I can't remember what the name of it, a concept article, that's what it was. In 2005, we included this in there. And that was nice because this was published just before this paper came out by Adam Heller and his postdoc Mano. They developed a biomolecular, what did they call it? Bioelectrochemical motion driven by glucose oxidase, and I don't remember what the other enzyme was, but they had electromigration from one side to the other side, and that drove these objects moving in the opposite direction of the proton flux that they expected to see. So when they put an insulator between the two ends of their device, they did not get any electromigration. So we were settling on this idea of electrochemical or electrokinetic mechanisms for the motion of these objects. And so the velocity for a self-electrophoretic mechanism should scale the same as an external electrophoretic experiment. The velocity should scale as a function of the particle's zeta potential and the magnitude of the electric field. And that electric field can be written as the flux of the ions over the conductivity of the solution that it's dispersed in. So what that means is that as we increase the conductivity of this solution, then we would see a decrease in the velocity, or inverse is as we increase the parameter 1 over the conductivity, we should see an increase in the velocity. So these are platinum gold rods and 3% hydrogen peroxide in 3 millimolar sodium nitrate. And we're just seeing essentially Brownian motion there. And then if we have that in the absence of the sodium nitrate, we see a significant increase in the velocity. So this was kind of a problem for the early catalytic nanomotors. If we added anything to these solutions, salts, surfactants, anything else, it would slow down the motion of these objects. But I think we pretty conclusively showed that the mechanism for these rods is, rod motion is due to, at least in part and primarily, electrokinetic behavior. And that was one of the tricky things about this, is that we could never, I mean, this is really kind of the scientific method in action, right? You have an observation, you propose a hypothesis, and then you design some experiments to test that hypothesis. And then you draw conclusions based on those results. And do those results confirm or rule out your hypothesis? And so we had some confirming evidence, but it became really difficult to find evidence that would rule out other potential mechanisms. And so I remember getting a little cautious about how we approached that when we published some of the later papers. But from motors to so we have these motors. A suspended motor can move itself, and that was published in JACS in 2004. And then the immobilized motor moves surrounding fluid. So if you take these, sort of like the inverse of the Howard Berg experiment, you immobilize the moving things on surfaces, well, then what will happen? And so in this case, we get fluid migration from one region to another region. And then my colleague, Tim Klein, talked about that in a JACS communication in 2005. So we wanted to confirm that and he was doing silver and gold because silver was easier to deposit. So we were looking at platinum systems. And so a platinum gold motor, you can think about the oxidation happening on the platinum side, the reduction happening on the gold side, and then some electron flux inside the wire. So how do you measure the current in a wire? Well, you could put an insulator there, and then you could get some really small contacts to contact the platinum and the gold, and then you could measure that the current that would go through the external circuit, so we can measure it as opposed to going through the rod itself. But it turns out that's a challenging experiment. So what we did instead is we just made a really big platinum section and a really big gold section, and we exposed that to hydrogen peroxide. We observed that we could get really high current densities from 3% hydrogen peroxide in this kind of an interdigitated platinum gold electrode setup. So we're just measuring the current from the platinum to the gold in the presence of hydrogen peroxide. And the predicted electric field is like 13 volts per centimeter, which is significant and is enough to drive electrochemical behavior. And so, we made some pumps also. This is the control experiment with the applied electric field, where we've got an electric field on the order of 18 volts per centimeter. And we're looking at the motion of these objects in the plane of the electrode. And then they get kicked up and they get kicked up into solution. But that initial, I'm not going to restart this video in case it crashes again. So we did the same kind of thing. Instead of an externally applied field, a internally generated, catalytically generated electric field on the order of 13 volts per centimeter. So the idea was that we would start off without the connection and then make the connection between the platinum and gold, allow electrons to flow in the external circuit and the protons to flow in the solution adjacent to these platinum gold structures. So once we flip that switch, you'll see the migration of things from the platinum to the gold, and then they get kicked up out of the plane as you start generating these sort of eddy flows of solution due to the electroosmotic behavior. I'm not going to talk too much about this, but this is just kind of how we interpreted those results a little bit more. But we did settle on this electrokinetic mechanism and finally published this in 2006. I remember when we had some concerns about this mechanism, we're like, okay, well, if the mechanism we published is wrong, well, what do we do? And the advice from my committee is, well, then you need to be the one to show why it's wrong and make the correction. And so, so we followed up that paper with this electrokinetic mechanism. Since then, we've had a lot of fun. So this was a picture, I think this was for Chemical and Engineering News. And so we see a couple, Jeff Kaczmark, Vin Crespi, Tom Maluk, of course, and Ayusman. This is Yan Kao, who did some early atomic force microscopy measurements for that JACS paper. This is Paul Lamert or Lamer. And then Shamala was a grad student for Jeff Katchmark, and then Tim Klein and me, way back when we were all young. So a lot of things have happened since then. I mentioned the remote control of catalytic nanomotors, moving cargo around with catalytic nanomotors, settling down on a more consistent mechanism, which was also confirmed by Yang Wang in Tom's group. Chemotaxis, this was Eve's work, or Ying Hong did this PRL paper showing chemotaxis, or the collection of catalytic nanomotors at high concentrations of hydrogen peroxide, changing the different geometries of these. And let's see, making some micropumps. One of the things that plagued us was trying to find some other reaction that could do this. And so we used platinum or palladium and hydrazine. Mike Eibel, who came towards the end of my time at Penn State. And then, you've arrived, right? When this is on the cover of Scientific American. So this was a really nice feature. I think this was 2009 when this came out. Now, it's important to point out that we weren't the only ones having fun, and there was a lot of other interest spurred on by that initial paper. Ben Faringa published a couple of papers. This one was this immobilized manganese catalyst on these glass shards, and he showed the bubble propulsion of these objects. He also did something similar using enzymes. So they tethered enzymes to nanotubes. And again, this is bubble propulsion due to the catalytic generation of oxygen, in this case, enzyme catalyzed. And then Joseph Wang came onto the scene, I think around 2008, where he said, well, you know what, I think I can make these faster. So he started playing around with other kinds of alloys and made things that were 150 microns per second. I think there's some other stuff at 200 microns per second, which is crazy fast compared to what we were doing early on. Jonathan House this is Jones' group, right? That they did this, and so they were calling this self-diffusiophoresis. I don't think that they distinguished it from auto-electrophoresis at the time, but due to asymmetry of platinum, that was, I remember thinking that it could still be self-electrophoresis on there. But anyway, lots of people having fun with this, and then this inspired some other kinds of motion. So not catalytically, not the auto-electrophoretic or auto-diffusiophoretic mechanisms, but artificial flagella started to appear. So some motion that's driven by actuating this artificial flagella magnetically. The tubular nanomotors came on 2009, I think. Oliver Schmidt and Joseph Wang. I think Samuel Sanchez came onto the scene somewhere in here as well. Frechet and Zettel were doing some surface tension mediated conversion of light to work, where they're shining a thermally absorbent material with a laser and then driving the motion of objects that way. Then there's some non-catalytic strategies. So this is the Sen group. So you might be familiar with some of this work already. Platinum and copper, and the dissolution of copper coupled to the reduction of some brominated species generates electroosmotic flow, and so these things move, but not by catalytic reaction, as far as I understand. I guess except this is catalytic process here. And then this was some diodes, and then using an alternating electric field where these diodes rectify the current and use that to generate a potential difference on one side versus the other side, and they'll swim around. Also, that was miniaturized. I think this was Joseph Wang also, where they showed that under a uniform electric field, they could get semiconducting particles to rectify and swim in solution. And then this one I thought was a particularly interesting example. This was thermophoretic behavior, where particles that were coated with some magnetic material could locally heat up in the presence of an alternating magnetic field. And then they showed the motion of this. So the coated side is pushing the object through solution. This is kind of, and then at this point, an awful lot of things started happening, and it became difficult to keep up. And if not for some of those micro and nanomotors meetings, 2016 and 2014, I don't know that I would know nearly as much about what's going on here, but it really kind of took off. And the problem with giving a talk like this is that inevitably I'm going to leave something important out, and that certainly wasn't my intention. But you know, we're still thinking about these challenges and opportunities for nanomachines. Is what kinds of mechanisms can we get to move things? What kinds of fuels can we use, and what kind of environmental compatibility will those mechanisms allow for? I know there's been some ways to move micro objects in biological solutions and in environmental solutions too. And then some of the other questions are, how do you control them? And we demonstrated controlling with magnetic fields, and then taxis, and then also using light to control those, and concentration gradients. But then some of the important questions moving forward are, what kinds of things do you want them to do besides move? If you just want to move matter, there are lots of ways to do that. Is there something else that you can get nanomachines to do? Can you start to incorporate other kinds of functionality to that? And I think were undergraduates in my lab who did a great job of you know, they were picking out particle trajectories by hand at the time. We didn't have any motion analysis. A lot of other people in the group who helped push this forward, and some people that Tom will remember, Nina Kovchakova, Sarah St. Angelo helped us out on some microscopy, and Yan, and then Paul Lammert, Yang Wang, and Tom Larrabee did some atomic force microscopy that was related to this. And then catalytic nanomotors, if you go into Web of Science, you'll see lots of publications and starting to come down. But I think that this is sort of artificial, because catalytic nanomotors seems to have morphed or been absorbed by this larger idea of active matter. So thanks for taking the time to listen to this. I'd be happy to take some questions. But you know, I guess one of my themes is inspiration from the past. There's still a lot there to be inspired by, and then looking to the future and seeing what else can happen with catalytic nanomotors. So thanks, guys.
Okay, thank you, Professor. Thank you for your wonderful talk.